the Institute for Health Research at Kaiser Permanente of Colorado. He's also an associate clinical professor in the Department of Epidemiology here at the Colorado School of Public Health. His research focuses on vaccine safety, vaccine acceptance, substance use, and overdose prevention. His background includes a Master's of Science in Biostatistics from the University of Massachusetts Amherst, and he completed his doctoral training in epidemiology at the Colorado School of Public Health. He's a principal investigator for the Vaccine Safety Datalink, which is a nationwide Centers for Disease Control funded project that examines the safety of vaccines and leads research efforts to develop risk communication tools to reduce parental vaccination concerns. He conducts observational studies to assess the risks of opioid use and interventions to minimize overdose risk. So welcome, Jason. Thank you so much.
Thank you. So I'll get started. During the talk, it's easier if I shut off the camera so it doesn't freeze, but then I'll turn it back on for questions. That's okay. Okay, so today I'm going to give a talk on vaccine safety monitoring in the United States with an example from COVID. I'd like to acknowledge Tom Shimabukuro, who is my colleague at the Immunization Safety Office at the CDC, who is a collaborator on this work. The objectives, I'll describe the aims of the CDC vaccine safety monitoring programs. I'll describe the main systems that CDC uses, including the VSafe, Vaccine Adverse Event Reporting System, and the Vaccine Safety Datalink. And then I'll describe some data supporting associations between COVID-19 vaccination and selected adverse event, focusing primarily on the mRNA COVID vaccines and myocarditis. Okay, so here's a slide of vaccine development, the basic research, discovery, and preclinical phases. So the first steps in vaccine development involve laboratory and animal studies. There's an exploratory stage involving basic laboratory research, typically lasting 2 to 4 years, where scientists try and find synthetic or natural antigens that could prevent disease. This includes weakened viruses or bacteria, weakened bacterial toxins and virus-like particles. Then there is the preclinical stage, often involving tissue culture or animal studies to evaluate safety and immunogenicity of the candidate vaccine. And these studies really just sort of give an idea of the cellular responses that might be expected in humans. And they also provide some insight into the safety of a starting dose for the trials, as well as a safe method for administering the vaccine. And sort of at this stage, a company who develops a candidate vaccine that demonstrates a desired immune response, they submit an investigational new drug, or an IND, to the FDA, which then summarizes all of the data and describes the proposed clinical protocol. And if the FDA approves the NDI application, the vaccine undergoes 3 phases of clinical testing. So as you can see on this figure, phase 1 is small. It's usually less than 100 participants. The objective is to assess dosing and safety and determine the type and extent of the immune response. And it tends to look for immediate reaction. So it's more about the tolerability of the vaccine. And then if this is promising, the vaccine is tested in a phase 2 clinical trial, which usually involves a few hundred participants. And the goals are to fine-tune the dosing, evaluate the method of delivery, and assess safety. And this is a randomized study with a control group. Then after phase 2, there's a phase 3 trial, those tend to be much larger. They involve thousands of participants for vaccines. They're randomized, double-blind. The goal is to assess vaccine efficacy and safety, to be able to detect rare adverse events, if possible, that couldn't be detected in the first 2 phases. And then if the vaccine is proves to be safe and effective in a phase 3 trial, the vaccine developer will submit what's called a biologics license application, BLA, to the FDA, who will then make a decision on approval and licensure of the vaccine. And if approved, the FDA will continue to monitor the production of the vaccine, including safety and purity. And then after approval, the Advisory Committee on Immune Practices, or ACIP, which is a group of experts convened by the CDC, will make recommendations on the vaccine schedule, the age and high-risk groups eligible for vaccines, and will also continue to review the safety data. And at this point, this is where the phase 4 or post-approval monitoring comes in. It's also known as post-marketing safety surveillance. And these are population-based epidemiological assessments after the vaccine has been licensed in large populations of 7 million people, often using electronic health records and claims data to identify very rare and potentially serious unexpected adverse events that couldn't be detected in the trials. And this is where the rest of my talk will focus on the post-marketing safety in the US. So what's the rationale for conducting post-licensure or post-authorization vaccine safety? Well, safety standards for vaccines are very high. They're given to healthy people. It's not like drugs, which tend to be given to people who are already ill. They also tend to be given to healthy children, primarily between birth and 24 months of age. So adverse events aren't tolerated, if you will, when you administer a drug or medication to healthy children. Also, the pre-licensure, pre-authorization trials are not designed to detect very rare events. Populations are often too small for many rare events. They often exclude special populations like pregnant persons and people with certain autoimmune conditions. They also aren't well situated to evaluate adverse events with delayed onset because the follow-up is too short. And so for that reason, we need to monitor vaccine safety in real-world observational environments. So what are the goals of a post-licensure safety monitoring program? Well, we try to identify rapidly unanticipated or rare events of clinical importance. We want to monitor changes in patterns for known adverse events. Continue to assess events that were identified in the trial. Assess safety in special populations, so including pregnant persons, people with rare autoimmune disorders, as I mentioned. And then also assess long-term risk of chronic conditions and conditions with insidious onsets. So for example, if you're looking at autoimmune or atopic diseases. So in the past, when parents were concerned that childhood vaccines might cause type 1 diabetes, for example, most vaccines are administered to children between birth and 2 years of age, yet diabetes tends to be diagnosed between 10 and 14 years of age. And this obviously can't be studied in a trial and would have to be conducted in an observational setting. And then lastly, the goal of a post-monitoring surveillance system is to provide timely and accurate data to stakeholders, as I mentioned before, including the Advisory Committee on Immune Practices. So before I describe the main monitoring systems in the US, I'll describe a concept called signal detection, as well as some other terms that we use in vaccine safety monitoring. So the Council for International Organizations of Medical Science defined a signal as information from one or more sources which suggest a new potentially causal association or a new aspect of a known association between an intervention and an event or set of related events, either adverse or beneficial, that is judged to be sufficient likelihood to justify a verificatory action. And so in practice, efforts focus on detecting signals for adverse events. And so here's the vaccine safety signal pathway that we use. So first, we monitor and analyze data to identify a signal. If the signal is identified, we conduct a formal hypothesis testing analysis to assess causality. Then if the signal is verified, we evaluate possible biologic mechanisms and develop prevention strategies. And such strategies often involve immunization recommendations and policies, like restricting vaccination to certain age groups, if an age group is at particularly high risk for an adverse event, or it may even involve pulling the vaccine from the market. And here's some terms that we use, and I think we're familiar with a lot of these, but we have an adverse event. We use this term all the time. And this is a medically attended event, identified usually with an ICD code, temporarily associated with vaccination that may or may not be caused by the vaccine. Then we use the term adverse reaction, and this is an adverse health event after vaccination that is likely caused by the vaccine. We have ICD-10 and 9 codes, which I think we're all familiar with. These are codes that are stored electronically in the electronic health record and claims data, and it represents a diagnosis for a healthcare encounter or for billing for a healthcare encounter. We use the term automated analysis, and so this is just an analysis of those electronic ICD codes. But then we also have medical chart confirmed cases, and this is a medical event in which the diagnosis or onset of the condition was confirmed by medical record review of the medical records, often by a clinician or trained medical records abstractor. We have incident case, standard definition, a medical event that represents the onset of the disease or condition. It's a diagnosis on a specific date. And then prevalent cases, which is a diagnosis representing documentation of an existing condition, which often can only be ascertained after the medical record review. We have biologically plausible risk intervals. And so this is the time period after vaccination in which an event can be attributed to vaccination. And then a statistical signal. We use this term a lot, a signal. And this comes from an automated analysis of ICD codes where the association exceeds a pre-specified statistical threshold. And so we call it a signal because it's suggestive of a vaccine safety problem, and it needs further assessment before conclusions can be drawn. And so that's a broad overview of the objectives of a vaccine safety monitoring system and some definitions of the terms that we use. And so now I'll describe the primary vaccine safety monitoring systems in the US, which are based out of the CDC. So within the CDC, the safety systems are governed by the Immunization Safety Office, or ISO. And I've been involved with one of these systems called the Vaccine Safety Datalink for the last 20 plus years. Now, the other systems are VAERS, vSafe, and CISA. And I won't be describing CISA this time. And for COVID, and then there's also the vSafe system, and I'll describe that. And that was set up specifically for COVID. So what is vSafe? So this was the promotional advertisement to encourage enrollment in vSafe, which is a smartphone-based active surveillance system. And you may remember this when you got your first COVID vaccine doses. And the CDC promoted vSafe very heavily early on in the COVID vaccination program. The idea was to create active surveillance systems that individuals could voluntarily enroll in after receiving a COVID vaccine dose. And the goal was to capture potential adverse events either early, before a person sought medical care, or events for which people typically didn't seek medical care for. So it was set up to supplement the existing vaccine safety surveillance systems. So vSafe was an active monitoring system as opposed to passive. So vaccinees registered on their smartphones. Children 15 years or younger could have been added through their parents' account. And it's considered active where text messages were sent to vaccinees to complete surveys. And vaccinees could access their accounts online as well. The online surveys were brief. The objective was to collect data on these non-medically attended events, so non-serious events primarily. And it collected symptom and long-term sequelae as well that may not be captured in medical records or claims. And it was also available in a variety of languages. And here's just an example of the data compiled by VSafe. It was fairly simple but informative. In this figure, the first row was for vaccinees younger than 65 in the bottom, or older, and the bottom was for younger. And the y-axis was the proportion of vaccinees, and then the x-axis was for the days following vaccination, starting with day 0. And the bars just represented the proportions of mild adverse events: injection site pain, fatigue, headache, chills. And again, the purpose of VSafe was really twofold. It was one, to be able to quickly identify signals for possible adverse events that perhaps vaccinees didn't seek care for, and two, to sort of just really instill confidence in the public that we were actively monitoring the safety of COVID vaccines. So the other system is called the Vaccine Adverse Event Reporting System, or VAERS. And the Vaccine Adverse Event Reporting System, or VAERS, was established in 1990, and it's currently co-managed by the FDA and CDC. It's a passive surveillance system that accepts reports of possible adverse events following vaccination. So anyone can submit a report to VAERS. That includes providers, patients, caregivers, manufacturers, anybody. Some key strengths: it can be used to rapidly detect potential vaccine safety problems. It can be used to detect rare events, but it has some very significant limitations. It's passive, so there's no denominator. The quality with which events are reported is inconsistent, and there isn't always complete information. It's subjected to reporting biases, and therefore you cannot use VAERS data to establish causality. And you do see, particularly with COVID, we saw a lot of individuals who sort of misused, bordering on abused this VAERS data to sort of claim that the vaccine was causing some serious health issues. This cartoon really just represents the timeline from vaccination to a submitted VAERS report. So the individual receives the vaccine, and then at some point within the first few weeks, the vaccinee experiences symptoms. And then at some point after the symptoms, they submit a report to VAERS. And so this event may or may not be related to vaccination. And this just reemphasizes that VAERS is a passive surveillance system of events reported as occurring just sometime after vaccination. There really isn't a denominator. There is not a denominator for vaccinees, nor is there an unvaccinated control group. So again, the data can only be used to generate hypotheses, not to test them. And this is just a screenshot of the VAERS form. It's fairly lengthy, and individuals can just fill out as much of it as they'd like and submit it to the CDC. This is just a 2x2 table of the figure I showed you. I know I'm sort of beating a dead horse now, but I think it's really important to understand that these VAERS data cannot be used to establish causation. So this is the 2x2 table that we're all so very familiar with. The VAERS data just gives you that gray shaded box. And so it can only be used to generate hypotheses. The other system that I mentioned I've been involved with is the Vaccine Safety Datalink. It is a collaborative project between the CDC and 12 integrated healthcare organizations across the US, so it's got good geographic representation. There's data on about 15 million people per year, electronic health records, and we use the VSD to conduct rigorous vaccine safety studies and near real-time monitoring. So the VSD data, we call it a linked database where each unique patient receives a random study ID that links their membership enrollment information from within their health system with demographics and ICD and procedure codes for medical encounters in the outpatient, emergency department, and inpatient settings. And these data are largely derived from electronic health records, as well as claims from inpatient encounters among the health systems that don't own their own hospitals. Mostly electronic health record data with access to the medical records from manual chart. We create sort of two sets of data for the VSD, starting with what we call our annual cycle files. So these are yearly files derived from the EHR and claims. They're longitudinal, dating back to 1981. They're harmonized based on a data dictionary. The data files, as I mentioned, are linked by unique study IDs, and we use the cycle files for sort of traditional retrospective observational studies. So here are the studies. The first is a cohort, which we're all familiar with. We use two sort of more novel designs called a risk interval and a self-control case series. We typically do for a cohort, a matched cohort, or vaccinated or matched to unvaccinated by age of the date of vaccination. We do this because vaccines tend to be given at specific ages, and some may be given seasonally, like flu. So there are secular trends and time varying factors that have to be accounted for. And with this design, we calculate the incidence of events in a pre-specified risk window, which is a time period in which an event can be biologically attributed to vaccination, and then compare it to the incidence in the exposed time outside of the risk window. And sometimes we truncate the cohort design where we just use the risk period in the exposed or vaccinated and compare it to unvaccinated time of the same length in the unexposed. And we do this if we're particularly concerned about time varying confounding, because there'll be less follow-up to worry about. Also, we may need the information quickly. So with this design, you don't have to wait as long to conduct the analysis. And if our sample size is large enough, we don't need all of the follow-up time to calculate stable rates. And then we use a risk interval analysis. It's a vaccinated cohort. So only vaccinated individuals are included. And the incidence in the risk period is compared to the incidence in the control period. So we do this because we're concerned about confounding by indication or contraindication. So for childhood vaccines, almost all of the children receive them because they're considered mandatory for daycare and school. So there are a few unvaccinated controls available, and the unvaccinated may be inherently different than the vaccinated by unmeasured confounding variables. Or the vaccines may be indicated for high-risk individuals, like COVID was for a while, which can lead to bias if the vaccinated are compared to the unvaccinated. And next is the self-controlled case series method. It's a case-only analysis that really revolutionized the field of vaccine safety. We use a self-control. I would say we use self-controlled designs extensively in the VSD. So only cases. So people who have the event of interest over a pre-specified observation period are included in the analysis, and time periods before and after vaccination are compared. So it's only cases. And because time periods within cases are being compared, it implicitly controls for unmeasured fixed confounding variables, meaning variables that do not change over time. And because it's a case-only analysis, the sample size is a small fraction of the entire cohort. And it's been shown that the SCCS is about as powerful as the entire cohort when the event is rare and the risk is high. So I should also add too that this method is primarily used for acute exposures and acute self-limiting events. It does not work very well for chronic exposures and chronic outcomes. The design, however, is susceptible to time measuring confounding. So one of the assumptions as though that background rate is stable over time if you don't control for it. And that can be a pretty limiting assumption in the sense that, if there are seasonal distributions to an outcome of interest, you could see something like this where in the control period, the background rate is higher than in the risk period. And even if there is an association between the vaccine and the outcome, if you fail to sort of account for that time varying variable, it could create a bias. We could say there is no effect when in fact there really is. And controlling for time varying confounding in a self-controlled design is challenging. Okay, so we use those designs primarily for some traditional retrospective analyses, but then we also create what are called dynamic data files, and these are for newly licensed and seasonal vaccines. And for the DDF, the data are updated on a weekly basis. So a cohort is created each week and followed longitudinally. And then the data are accessed using what we call a dynamic data model, which allows each site to maintain local control over their data. So this is a figure of the distributed data model, and the dynamic data files are accessed and analyzed through what we call the distributed data model. And there's a hub, which is at Northern California Kaiser, behind their firewall. And then each VSD site houses their data files behind their respective firewall. And the sites and the VSD can submit SAS programs to query the data by submitting SAS programs to the hub. And then from the hub, the programs are relayed to the sites. Sites can review the programs, which are then run on their respective data sets. And a subset of the data or the results are sent back to the hub, which then can be retrieved or downloaded by either a site analyst or the CDC. And so each site retains local control over their data. And then using the distributed data model, we conduct what are called rapid cycle analyses. And so we use these data from the distributed data model. We compare a number of outcomes observed versus outcomes expected. We include methods for sequential testing. Since we look every week, we want to decrease our type 1 error. It's designed to detect signals, and then when a signal occurs, we conduct a series of more formal evaluations to confirm that signal. We also do chart review of the cases to try and exclude non-cases and confirm the signal. And typically for the RCA analysis, we use a self-controlled risk interval method, which is a hybrid between the risk interval and self-controlled case series. And it's like the risk interval in that you start with a cohort of vaccinated individuals and risk and control windows. But it's like the SCCS or self-controlled because only cases are included in the analysis. And we use this for rapid cycle analyses of newly licensed and seasonal vaccines because it gives us an assessment of vaccine uptake as the vaccines rolled out. It also addresses unmeasured fixed confounding. And the analyses can be done quickly because the follow-up tends to be short and only cases are included in the analysis. And so that's an overview of the VSD. And here are just some resources that if anyone's interested in, they can look at on the CDC website. So I'll walk through a specific example of how VAERS and VSD worked together during the COVID pandemic after the rollout of the COVID vaccines. So once the CDC vaccines received emergency use authorization from the FDA, we set up a rapid cycle analysis in the Vaccine Safety Datalink. The time period was between December 14, 2020 and June 26, 2021. It included 6.2 million individuals and about 12 million doses of the COVID vaccine, mRNA vaccine. The mean age of the cohort was 29 years, 54% female, and we looked at 23 pre-specified outcomes, and this is for the weekly RCA analysis. So here's a list of the outcomes, and these outcomes were based on known, potentially serious adverse events, either from other vaccines that we studied in the past, events examined in the COVID vaccine clinical trials, and events that we knew were identifiable with a certain degree of accuracy in the electronic health record databases. And this outcome list was compiled by VSD investigators in collaboration with partners with the CDC, FDA, and Department of Defense. We used a fairly complicated design for this. It was a hybrid self-controlled risk interval. And so for the vaccinees, we looked at outcomes occurring in a 20-day risk window following vaccination. And then the comparison window was days 22 to 42 in vaccinees who were matched or anchored by age, sex, race, ethnicity, and VSD site. And so why did we do it this way? So if we'd done a traditional self-controlled analysis, we would have had to wait 42 days after vaccination to start accruing person time in a post-vaccination window. And this would have delayed the analysis. And at the time, we felt like we needed to get results out as soon as possible once the vaccine was rolled out. And we didn't want to use unvaccinated comparators because they would have been very different than the vaccinees by underlying risk conditions, particularly early in the vaccination program. And so we went with this sort of hybrid design. And initially, these were the results. And we used, if you look in the second to last column, that was the adjusted p-value based on the sequential testing, because we were looking every week, so we had to adjust the p-value to trigger a signal. And none of the outcomes triggered a signal. And I highlighted myocarditis because initially we didn't see anything. And so at this point, we concluded that there's nothing to be concerned about with the vaccine. And so what is myocarditis? I'll talk about that in a second. I'll talk about this before I go into the VAERS data. So myocarditis is inflammation of the heart muscle. It's most commonly caused by a virus, and it can occur after COVID infection. The clinical presentation, of course, is variable. The symptoms include chest pain, shortness of breath, palpitations. And what happened was we saw an increased reporting of myocarditis and pericarditis after COVID vaccination in the spring of 21. It was first noted by the US military. And so the VAERS data, in contrast to so these were done concurrently. We were doing rapid cycle analysis, and then VAERS, of course, was collecting reports. And in contrast to the VSD, VAERS did see a signal for myocarditis. And this paper came out and concluded that there does appear to be an increased risk, and it appears to be coming after the second COVID dose in adolescent males and young men. And this is a table from that paper of the VAERS data. The signal was greatest in males 16 to 17 years of age with 1.3 excess cases of myocarditis in a 7-day risk interval following vaccination. And here's a graphical display of those reported cases. And you can see there is, so on the left is the Pfizer mRNA, and then the right is the Moderna. And there is clearly a clustering of doses in those first 4 days following vaccination, primarily after that second dose. So after seeing this, we had to go back to the VSD data, and we restricted the data to the age group that the VAERS data showed a signal in. And sure enough, we did see this spike in the same time period. And then we conducted an analysis limited to that age group in a shorter day risk interval. And we saw a very high relative risk. And I think this is interesting. It shows how the two systems can work together. It also shows the limitations of, I think, the VSD. We didn't see this because we had all the ages lumped together. And the signal it was very specifically in this age group, primarily in males, and in a shorter risk window, and it was being obscured with data together. But then after the VAERS signal came out, and this is how the systems are meant to complement each other. VAERS generates a hypothesis, and then VSD, we can test it. And clearly there was a large increased risk of myocarditis in the 7 days following this, primarily the second dose in adolescent young adult males. So this prompted a follow-up analysis that said, okay, well, we now know, and other systems across the world were finding this as well. We want to know, well, there's two mRNA vaccines, is one riskier than the other? So we did a follow-up, so we called it a head-to-head analysis, where we compared the Moderna to the Pfizer. And then sure enough, there was an interaction. The risk following the Moderna was greater than the risk following the Pfizer by a certain degree. So the next question, though, so it looked pretty clearly that there was this increased risk. There was an excess of myocarditis cases following COVID vaccines and what we would expect. And so we wanted to know, is vaccine-induced myocarditis different than, say, viral-induced myocarditis? So there was follow-up of the cases. In the medical record review of the follow-up of the cases, it looked like all the symptoms had resided by the time a case was followed up. But the CDC has been conducting ongoing sort of follow-up of some of these cases. They haven't finished everything, but the preliminary findings suggest most patients report no impact on the quality of life, or missing school or work. And most indicated that the patient was probably fully, or I should say partially or fully recovered. And again, they're continuing to monitor this. There is, I will say, however, some new data that's concerning out of Australia that surveyed patients who had myocarditis after COVID vaccination, and 6 months later, they are still reporting symptoms. And so this is ongoing, and I think these results could affect future recommendations for upcoming well, the COVID vaccine is being recommended in the same manner as the influenza vaccine. It's seasonal, but it's possible that this season we see some changes to the recommendations. So these results translated directly into policy decisions. A rare risk of myocarditis was confirmed following the mRNA vaccines. It was confined especially to males 12 to 39 years, and the CDC concluded that they should be aware of the risks following the receipt of these vaccines. They concluded an 8-week interval between the first and second primary doses may be optimal for some people, as it may reduce the small risk of myocarditis. And then there's a precaution that said until additional safety data are available, experts advise that people who develop myocarditis after a dose of Moderna or Pfizer should not receive a subsequent dose. So in conclusion, there is some strong complementary vaccine monitoring systems that are in place to rapidly detect and assess potential safety concerns. COVID vaccines are being administered under the most intensive vaccine safety monitoring effort in U.S. history. Rapid assessment during the pandemic has informed policy and clinical considerations in near real time. And I think that the experience during the COVID-19 pandemic demonstrated that CDC vaccine safety surveillance systems can adapt to meet public health needs. And so I can end it there to see if folks have questions. A question?
Jason, I have a question. Thank you for
Sure.
your very, very interesting and clear talk. And maybe you covered this, but you mentioned in the very end where you're talking about myocarditis and how one system didn't pick it up and the other one did. Is there is there a recommendation that the analysis in the first system try to pick up more age-related or whatever types of effects, or do you still think that this is sort of the way to go? Working them together. Does that make sense?
Yeah, I think we for sure learned something. I think it's difficult to sort of identify risks in subgroups because we do tend to look at so many different outcomes. I showed you that.
Right.
It's it's interesting because we sort of say we're gonna look at all of these different outcomes. We're gonna test every week. We're gonna set ourselves up for finding something. It's this sort of better safe than sorry approach. But then at the same time, it feels like it's a bit of a paradox. By lumping it all together, we might actually miss something that's more specific. And so to your point, we have sort of played with looking at different methods like a tree scan, like a more, what do they call it, machine learning type of approach that would sort of mine the data in a little bit less of a targeted way to see if we could identify risks for adverse events in subgroups that perhaps we didn't specify a priority.
Interesting. Thank you.
Uh, Hi there. Great talk, Jason. I'm wondering, most of my research is in the meta research side of things and sort of trials as well. And I do study sort of methods for harms as well in that context. I guess, what do you see is the best opportunity or maybe the best way that these kinds of data could be used to inform future or ongoing in the sort of a pandemic setting when everybody's trying trials and you're also trying to collect as much data as you can on the harms of things, and trials often don't do a great job of that to start with, sort of, I guess, complementing how the trials should be set up or could be set up, and then also complementing what the trials find with the systematic review of the literature once the evidence has started to come together a little bit more.
And so I will say that modern trials for vaccines have adapted to this to a certain degree in that they tend to be much, much larger than they used to be because of this concern of potential rare serious adverse events. So this started back to when I first started working at the VSD in the 20s, just 1999, 2000. Some of you might have heard of the rotavirus vaccine and intussusception. Some of you might be too young to remember that, but the initial rotavirus vaccine generated a signal for a condition called intussusception, which is where the infant's intestine starts to telescope on itself, and it creates a blockage, and it's 100% treatable, but if it's not treated, the tissue can get necrotic and it can be fatal. It's considered a serious. It's treated with a barium enema to sort of untangle the. And I remember I had just started on this project, and there was calls discussing this potential signal, and most people on the clinicians on the call were like, "It doesn't make sense that the vaccine would cause this." It wasn't seen in the trial, which just had less than 1,000 people, but it's a very, very rare event. It turned out that association was confirmed in various systems, including the VSD, that the rotavirus vaccine was causing intussusception, and the vaccine was pulled. It was pulled from the market. They then developed another one, which is two more rotavirus vaccines, which we're using now, but those were done with trials of well over 10,000 infants. And so the trials have adapted in the sense that there's now set up, and the COVID vaccine trials were huge too. And they're particularly set up to have very large, because again, they're given to healthy individuals. And when you give something to a healthy individual, an adverse event isn't tolerated the same as when you give a drug to someone who's already sick. And so the trials tend to be much, much larger. I think, and the other thing that these systems have adapted is there is this, initially when we did this project, most of our studies were retrospective. They were a little slower. They took a few years to complete. We were methodical, calculated about how we did them. And then we set up that rapid time surveillance that I described. And that was done too, because if the vaccine is causing something serious, we want to identify it sooner than later. And so we do these weekly updates. We're able to do that because we have the electronic health record. It's not claims where we might go to market scan or opt-in where you buy the data and the data is already a year old. It's done as it's being collected within the health systems. And so that's why the VSD is set up that way. It's multiple health systems with electronic health records that then feed weekly updates of cohorts that we can analyze on an ongoing basis.
And Jason, I have a question related to your answers to Riyadh's question, which is, I sort of thinking about the whole signal detecting. You start with something, you hypothesize, or maybe during the clinical trial, you observe certain events, and those could be pre-specified for further surveillance. But vast majority of the adverse events, my guesses, are unspecified at the very beginning. So you sort of have to go through this process from known unknown to unknown unknown, and from which point where those non-specified predefined events become things that you have to put in a lot of effort to either estimate the risk or monitor them more closely. And how do all these systems play together to make that transition from starting with something you may anticipate or may expect to things you have never thought about that could have happened?
Yeah, I mean that's something we struggle with all the time is, how do we do this in a way where we're able to detect something completely unanticipated versus just seeing a lot of noise, right? Having a bunch of false positives. And it's a difficult line to sort of, balance, in the sense that if you see all these false positives, one, they're hard to confirm. It's a ton of work for every false positive to sort of do all the chart review, reanalyze. It's often just not feasible with resources. And two, it could create a lot of concern. If you see these signals for terrible outcomes, it's not always helpful to sort of have them create all this concern and then later follow up and say, oh, by the way, nothing's there. So again, we've been sort of struggling with a good way to sort of go about that. One of the things we're playing with now is using what's called the tree scan statistic, which you might be familiar with. And it's considered sort of a controlled way of data mining. And that's sort of in the preliminary phases, but that's one way in which we're sort of trying to figure out if we can use this method to identify things that are unanticipated. And, we get criticized often because we have these lists and people who are just inherently think that vaccines are not safe criticize us by saying, yeah, but there's stuff that's not on this list that it could be causing it, you're not fine. So it's just a dilemma. Yeah.
Any
I mean,
more
I do think
questions?
I was going to say real quickly, I think that's a really good area for further research now, is what are some good methods that can identify these unanticipated events and minimize false positive signals.
Great. Any other questions for Jason? Well, thank you, Jason, so much for presenting to us on your work. Super interesting.
Thank you.
Thanks, Jason.
Bye.
Bye.
Thank you.